Thank you for joining me. My topic today is the cell-penetrating peptides. My main focus will be the peptide design, peptide synthesis, and its applications. First, let me briefly introduce Lifetein. Lifetein was founded in 2008. We have been in the peptide industry for more than 10 years. We specialize in peptide synthesis, chemical synthesis, antibody production, and protein services. Our main focus is the peptide synthesis. However, over the years we have expanded to other protein-related areas like proteins, antibody services, and products. So let us quickly get into the topic, cell-penetrating peptides. What is a cell-penetrating peptide, or CPP? From the definition, CPP is a short peptide that can be about 4 to 40 amino acids in length. The short peptide can enter the cell membrane and then deliver bioactive cargoes. CPPs can also be used to deliver bioactive cargoes like siRNAs, DNA, polypeptides, liposomes, nanoparticles, and others into the cell for therapeutic or experimental purposes. There are a few popular models for CPP entry. Number 1, the inverted micelle model. The CPPs are positively charged. They interact with the negatively charged phospholipids in the membrane. Number 2, direct entry or direct translocation. For example, sequences with multiple arginine residues can cause a short-term membrane cytolysis and enter the cells directly. Number 3, by the traditional method of endocytosis. I will not talk about the details on this one. Here are a few examples of the CPP. The most famous examples are HIV TAT sequences. The TAT peptide is arginine-rich and can directly penetrate the plasma membrane and stabilize DNA. Another example is the arginine-rich peptide R8 or R9. We can add stearic acid to the N-terminus. Stearic acid is a saturated fatty acid with an 18-carbon chain. If you would like to do live cell imaging, we can add fluorescent dyes such as FITC, Alexafluor, or Cy-dye to the N-terminus or C-terminus. I will get to the details. CPPs can enter the regular cell membranes. Some other peptides are tissue-targeting peptides. For example, this brain-homing peptide can cross the blood-brain barrier. Other peptides can cross the skin as transdermal peptides, target heart tissues as cardiac-targeting peptides, and nuclear localization signal peptides. On this slide, I will talk more about the peptide design. There are many ways to make the peptide permeable. In the case of DNA or RNA, you can simply mix the CPPs with oligos. Many transfection reagents are using this mechanism. Simply put, DNA is negatively charged and the peptide is positively charged. If you mix them together, they will form small micelles for cell penetration. However, most of our work is put into the CPPs and your target together by covalent links. For this example, we put 8 arginine residues at the N-terminus of your peptide. A linker called AHX is added as a spacer. Some users prefer no spacers. It seems that both work well for the purpose. The 8 arginine residues can be put at the C-terminus as well. According to the feedback from our users, most of the N-terminal CPP work well. A few work as well for the C-terminus conjugation, though it really depends on the project. This example is the NPYS linker modification. The cysteine residue is added to your peptide. We conjugate 2 sequences together to form a disulfide bond. This is especially useful for cancer studies. Cancer cells have a lower pH of 6.7 to 7.1. Normal cells have a higher pH of 7.4. Under the acidic environment, the disulfide bond can be cleaved. If your target peptide is a cancer drug candidate, the CPP can introduce the drug cargo to the cancer cell and release the target within the cell. These disulfide-based prodrugs are important for cancer therapy. If the cysteine is not available for your case, we can add a compound called lysine azide. This method needs click chemistry. There are 2 kinds of click chemistry, the one with copper as the catalyst and the one without. The preferred method is the copper-free click chemistry. It is called DBCO and azide reactions. The final conjugate will have a large linker. Many scientists have concerns about this bulky size of the linker. However, some drugs contain bulky linkers without issues or side effects. Back to slide 9. Let us go back to the sequence. The design does not have to be this way. The lysine azide can be any place in the sequence. If you have a head-to-tail cyclic peptide, you can add the lysine azide in the middle. The final product will be like a lollipop, with the CPP as the tail. If the N-terminus is very important to you, you can add the azide to the C-terminus. Let us move to the other scenarios. If you would like to track the peptides in live cells, fluorescent dyes can be added. We can do FITC, FAM, Cy3, Cy5, Cy7, and Alexafluor. This design will give direct evidence that your target is inside the cells. There is a different kind of peptide called a peptide nucleic acid, or PNA. It is a DNA or RNA analog. We can synthesize half as a peptide and the other half as a PNA. This structure is the one I just mentioned earlier. The cyclic tumor-targeting RGD peptide can be linked with an R8 cell-penetrating peptide to form a lollipop-shaped structure. So far, we have mentioned different ways to conjugate the cargo with cell-penetrating peptides. If your targets are nanoparticles or gold particles, our requirement is to have active groups like a thiol group or a free amine on it. We have to have the active groups react to the cell-penetrating peptides. It is the same requirements for small compounds. The last concept I would like to introduce is the antibody-drug conjugate. This concept is widely accepted in the antibody drug industry. There are three important components: an antibody, a cleavable linker, and the drug. Once the antibody binds to the target, the drug is released after the hydrolysis by protease. The same concept can be used for the peptides. For this concept, we need to screen the best drug candidate for cell entry. The CPP can be tumor-homing peptides, brain-homing peptides, or cardiac-targeting peptides, as I mentioned earlier. First, we need to modify the compound. It is better to have a free amine group in the compound. Then we can modify the amine group to an azide group. Afterwards, we can use click chemistry for the following conjugation. Lifetein produced a series of CPPs. They are ready to conjugate to your compounds for screening. So far, we have designed and produced more than 50 CPPs. Step 3 is conjugate the peptides with drug candidates. Once the CPP is conjugated with the drug compound using click chemistry, we can send the final product back to you for further screening. The purpose is to find the best drug delivery system. To summarize today's topic, I talked about cell-penetrating peptides with different cargos. As long as you have an active chemical group on the nanoparticles, compounds, or liposomes, we can conjugate the target to any peptide. That's all for today. Please don't hesitate to contact us by email or phone if you have any additional questions.